For more than a century, one specific object has sat at the intersection of religious devotion and hard scientific testing more than almost anything else in the world. The Shroud of Turin, a length of ancient linen bearing the faint image of a man's body, venerated by many as the actual burial cloth of Jesus Christ. In 2015, scientists finally applied modern DNA sequencing directly to particles trapped in its fibers. What they found didn't prove the cloth's origin one way or the other. It added a genuinely interesting, genuinely complicated new piece to a puzzle that remains, after well over a century of direct scientific testing, still not fully resolved. Let's get straight into what that DNA study actually found, and just as importantly, what the researchers who conducted it actually concluded from it. Because the real, careful scientific interpretation is considerably more measured than some popular retellings suggest. Stick around because this is a genuinely fascinating case study in how to read a real scientific finding responsibly, without either dismissing it or overselling it. Hit subscribe if deep, evidence-based investigations like this one are your thing. Let's start with what the Shroud of Turin actually is, because understanding the object itself is essential before getting into what the science has and hasn't established about it. The shroud is a rectangular piece of linen cloth, roughly 14 feet long and 3.5 feet wide, kept today in the Cathedral of St. John the Baptist in Turin, Italy. Viewed under ordinary light, the cloth shows a faint, brownish image of a human figure, front and back, showing what appear to be wounds consistent with a crucifixion, marks on the head, wounds at the wrists and feet, and lacerations across the back, consistent with a severe beating. The image itself has no detectable paint or pigment applied in a conventional sense. The discoloration appears to affect only the very outermost layer of the linen fibers, a detail that has made the image genuinely difficult for researchers to fully explain using known medieval artistic techniques. And it's one of the central reasons the object has remained a subject of serious scientific interest, rather than being dismissed outright. The cloth is stored under carefully controlled conditions inside a specially designed case, and it is displayed publicly only rarely, with expositions typically occurring at intervals of years or even decades, rather than on any regular schedule. Each public exposition has historically drawn enormous crowds, with recent expositions in Turin drawing well over 1 million visitors across the several weeks the cloth was on display, a detail worth keeping in mind for understanding just how many individual people have had some form of direct or close proximity contact with the object across its documented history. The shroud's documented historical record becomes clear starting around 1354, when it appears in the possession of a French knight named Geoffroy de Charny in the town of Liray, France. From there, its ownership and location are reasonably well documented across the following centuries, eventually arriving in Turin in 1578, where it has remained ever since. The Catholic Church has notably never issued an official declaration of the shroud's authenticity as a genuine burial cloth from the 1st century. Church officials have generally described it as an object worthy of veneration and reflection, regardless of its precise historical origin, a deliberately cautious position that leaves the specific scientific question of its age and origin open for ongoing research, rather than settled by religious authority. Pope John Paul II, for instance, referred to it publicly as an icon worthy of contemplation rather than making any explicit claim about its literal authenticity, a framing subsequent church leaders have generally maintained. The event that first drew serious modern scientific attention to the shroud occurred in 1898, when an Italian photographer named Secondo Pia was given rare permission to photograph it during a public exhibition. When Pia developed his photographic negative, he discovered something striking. The negative image appeared far more lifelike and detailed than the faint positive image visible on the cloth itself, an effect consistent with the original image on the cloth already behaving in some sense like a photographic negative. This finding, later confirmed by independent researchers, became one of the central scientific puzzles associated with the shroud. Since photography itself didn't exist as a technology anywhere near the medieval period most historians associate with the cloth's likely origin, raising a genuine, still debated question about exactly what physical or chemical process could have produced an image with these specific and negative-like properties. Over the following century, the shroud was subjected to an unusually extensive range of scientific testing: X-ray analysis, ultraviolet and infrared imaging, microscopic fiber analysis, and eventually, in 2015, direct genetic analysis of particulate matter recovered from the cloth's surface and from between its threads. It's worth being clear from the outset that this level of sustained scientific attention is genuinely unusual for any historical artifact, religious or otherwise, and it reflects real, legitimate scientific curiosity about an object whose specific method of image formation remains, even today, not fully and conclusively explained by any single proposed mechanism. a genuinely open technical question, separate from, and not equivalent to, a claim that the cloth's religious authenticity itself remains scientifically undetermined. Now, let's get into the DNA research specifically, because this is the study at the center of the story. And understanding exactly what the research team did, and exactly what they concluded, matters considerably more than the headline alone. In 2015, a research team based at the University of Padua in Italy, led by geneticist Gianni Barcaccia, undertook a detailed genetic analysis of particulate material collected from the shroud's surface and from material recovered during previous restoration work on the cloth. Their study was published in the peer-reviewed journal Scientific Reports, part of the Nature family of journals, meaning the research underwent standard peer review before publication, a genuinely important detail for evaluating how seriously to take its findings and its author's own stated conclusions. The specific collection methodology matters for understanding what this study could and couldn't reliably establish. Researchers working with this kind of trace genetic material, recovered from a surface handled and exhibited over many centuries, rather than from a sealed archaeological context, face genuinely difficult technical constraints that don't apply in the same way to, say, ancient DNA recovered from a carefully excavated burial site. Material recovered from a widely displayed, extensively conserved textile has been exposed to open air, repeated handling by conservators and clergy, and direct contact from countless individual visitors across public expositions spanning centuries, none of which was collected or preserved under anything resembling the controlled conditions ancient DNA researchers typically insist on when trying to recover a genuinely ancient, uncontaminated genetic signal from remains excavated directly from a sealed burial context. The research team used modern next-generation sequencing technology, capable of reading and reconstructing genetic information from extremely small, degraded quantities of DNA, to analyze the collected particulate matter. What they found was genuinely striking on its face. The genetic material recovered from the cloth showed evidence of multiple, geographically diverse human population signatures, rather than a single, clean, consistent genetic profile. Signatures consistent with populations from the Middle East, from South Asia, and from East Asia were all identified within the sampled material, alongside genetic material consistent with European populations as well. The study also identified plant-derived genetic material from a range of species, including some associated with regions outside Europe, adding a further layer of complexity to the overall genetic picture the researchers were working with. Here's the part that matters most for understanding this study responsibly, and it's the part that popular retellings of this research most often get wrong or leave out entirely. The research team's own conclusion, stated directly in their published paper, was not that this genetic diversity proved the cloth had traveled along ancient trade routes collecting genetic material from a diverse range of ancient populations across the centuries. Their conclusion was essentially the opposite, that this kind of genetically diverse, geographically scattered signature is exactly what you would expect to find on any object that had been extensively handled, touched, and contacted by large numbers of different people across many centuries of religious veneration, public exhibition, and repeated restoration work, particularly for an object as widely visited, handled, and physically worked on as the shroud has been throughout its well-documented history since the 14th century. This is a genuinely important scientific distinction, and it's worth explaining clearly why researchers studying ancient or historical DNA generally treat a highly mixed, geographically scattered genetic signature as a strong indicator of contamination, rather than as meaningful historical evidence. Ancient and historical DNA analysis is notoriously vulnerable to contamination from modern sources. A single touch, a stray skin cell, or even airborne particles from anyone who has handled an object can introduce genetic material entirely unrelated to that object's actual history. This is exactly why rigorous ancient DNA laboratories working with genuinely old, archaeologically excavated remains go to such extraordinary lengths to avoid contamination. Dedicated cleanroom facilities, full protective clothing for anyone handling samples, and strict protocols limiting who can even enter the physical space where extraction occurs, precautions developed specifically because researchers in this field learned, often the hard way in the field's earlier decades, just how easily a single contaminating touch can compromise an entire genetic analysis. Given how many hundreds of thousands of people have viewed, and in many documented cases directly touched or come into close physical contact with, the shroud across public expositions spanning more than 6 centuries, alongside multiple documented restoration efforts involving handling by conservators, researchers, and clergy from many different backgrounds. Finding a genetically mixed, geographically diverse signature on the cloth's surface is entirely consistent with, and arguably the expected outcome of, this kind of extensive, well-documented handling history, rather than being evidence the researchers themselves interpreted as pointing toward some specific ancient trade route or historical journey the cloth supposedly took. It's worth being direct about this because it's the single most important corrective to how this study sometimes gets described in less careful retellings. The Barkaccia team's own published conclusion explicitly favored the contamination explanation as the most scientifically responsible interpretation of their data, precisely because a modern object handled by millions of people across centuries would be expected to accumulate exactly this kind of genetically mixed surface signature, regardless of where that object originally came from or how old it actually is. The paper itself, read directly rather than through secondhand retelling, is notably cautious in its language throughout, consistently framing the genetic diversity found as a reflection of the cloth's complex handling history rather than as evidence bearing on any specific historical journey or itinerary, presenting the same data as direct evidence of a specific ancient journey along Silk Road trade routes without including the researcher's own considerably more cautious interpretation represents a real, meaningful misrepresentation of what the actual peer-reviewed publication concluded. It's worth noting, too, that the researchers themselves were reportedly measured and cautious in subsequent public comments about their findings, consistently declining to frame the diverse genetic signal as evidence supporting any particular historical narrative about the cloth's origin or travels, instead repeatedly emphasizing the contamination-driven explanation their own data most directly supported. That's not to say the study itself lacks any scientific value. It doesn't. Genuinely establishing that an artifact this extensively handled carries a highly mixed, contamination-consistent genetic signature is itself a real, useful scientific finding, relevant to understanding the object's documented handling history and useful for informing how future researchers might approach any further genetic testing of the cloth, including what kind of controls and precautions would be necessary to distinguish any potential genuine ancient signal from the extensive modern contamination clearly present in the samples collected. If anything, this study stands as a genuinely useful cautionary case study for the broader field of historical and forensic genetics, a clear, well-documented illustration of exactly why contamination has to be the default working hypothesis for any surface-collected genetic sample from a widely handled object, rather than assuming any detected signal necessarily reflects the object's own deep history. Let's also address the pollen research that frequently gets discussed alongside the DNA findings, because it deserves the same kind of careful scrutiny. Beginning in the 1970s, a Swiss criminologist named Max Frey conducted pollen analysis on tape samples taken from the Shroud, reporting that he had identified pollen from plant species native specifically to the Middle East, including some species found only in a narrow geographic band around Jerusalem. This finding has been widely cited by proponents of the Shroud's authenticity ever since, often presented as one of the strongest independent lines of physical evidence supporting a genuine Middle Eastern origin for the cloth. It's important to include a significant piece of context that responsible coverage of this specific claim needs to address directly. Max Frey's broader scientific credibility was seriously and publicly damaged later in his career, when he authenticated the forged Hitler diaries in the early 1980s, a fraud that was subsequently exposed and that led to serious, sustained professional criticism of his forensic methodology more broadly. That specific episode involved Frey confidently declaring a set of documents genuine that later, thorough forensic investigation proved to be an elaborate modern forgery, a serious professional failure that inevitably raised broader questions about the rigor of his forensic judgment generally, well beyond just the specific Hitler diaries case itself. This doesn't automatically invalidate his earlier Shroud pollen work specifically, but it's a genuinely relevant piece of context that responsible reporting on his findings should include, and it has led a considerable number of subsequent palynologists, researchers specializing in the scientific study of pollen, to treat his original Shroud sample collection and identification methodology with real, warranted skepticism, given documented irregularities in how his samples were collected, labeled, and later distributed for independent verification. Critics have specifically raised concerns about the chain of custody for Frey's original tape samples, about the lack of independent, blinded verification of his species identifications at the time he made them, and about the general difficulty of ruling out contamination or misidentification in pollen, samples collected using adhesive tape applied directly to a widely handled textile surface, rather than through more rigorously controlled sampling methods. Some later researchers have partially corroborated elements of his findings using independent sample sets, while others have raised serious methodological concerns that remain genuinely unresolved. The pollen evidence, in other words, is real, and it has been investigated by multiple researchers, but it remains considerably more scientifically contested and unsettled than confident popular retellings typically acknowledge, a genuinely open question within the specialized research community. Rather than the kind of settled, independently verified physical evidence, it's sometimes presented as in less careful popular coverage. Now let's address the radiocarbon dating, because this is the single piece of evidence most directly relevant to determining the Shroud's actual age, and it deserves to be presented accurately and with its proper scientific weight. In 1988, samples from the Shroud were independently tested by 3 separate radiocarbon dating laboratories at Oxford, Zurich, and Arizona using accelerator mass spectrometry, a well-established standard scientific dating technique that measures the ratio of a specific radioactive carbon isotope remaining in an organic sample to estimate how long ago the source material stopped absorbing new carbon from the atmosphere. All 3 laboratories, working independently of each other without coordinating their results in advance, returned closely consistent results, dating the cloth's linen fibers to a range between approximately 1260 and 1390 CE, a medieval origin, directly contradicting a 1st century origin. The fact that 3 separate laboratories, using the same general technique but operating independently, arrived at closely matching results is itself a genuinely important detail supporting the reliability of this specific test. Radiocarbon dating is one of the most extensively validated and widely used dating techniques in all of archaeological and historical science, applied successfully to date organic material across an enormous range of contexts worldwide, and the basic underlying physics and chemistry of the method are not seriously disputed within the broader scientific community. This remains, to this day, the single most direct, most rigorously controlled scientific test ever conducted on the Shroud's actual physical age, and it remains the primary piece of evidence most mainstream scientists and historians point to when assessing the cloth's likely origin. It's true that some researchers, most notably chemist Raymond Rogers in a 2005 paper, proposed that the specific sample, tested in 1988, may have come from a corner of the cloth that had been subject to an undocumented medieval repair or reweaving, potentially skewing the radiocarbon result younger than the cloth's true age, based on his analysis suggesting the specific tested corner showed chemical properties somewhat different from other parts of the cloth. This hypothesis remains a genuine, actively discussed topic within the small community of researchers focused specifically on Shroud studies, but it's important to be accurate about its actual standing within the broader scientific community. It remains a minority position, has not been independently confirmed through additional controlled radiocarbon testing of different verified sample locations, and has not displaced the 1988 dating as the scientific consensus regarding the cloth's most likely age. Several independent chemists and textile experts who have reviewed Rogers' specific evidence have raised their own methodological concerns about his analysis, and no follow-up radiocarbon testing using verified, uncontested sample locations has been conducted since 1988 to either confirm or resolve this specific dispute definitively, largely due to the Church's understandable reluctance to authorize further sample removal from an object of such significant religious and cultural importance. Responsible coverage of this topic should present the reweaving hypothesis as a genuine, still debated proposal worth mentioning, not as an established scientific correction that has overturned or discredited the original 3 laboratory radiocarbon result. There's also the question of the blood-like stains found on the cloth, another area where careful, accurate framing matters considerably. Testing has confirmed that the reddish stains on the Shroud, positioned at locations consistent with the wounds depicted in the image, do contain genuine blood components rather than paint or pigment, a real and interesting finding in its own right, and one that has been independently confirmed by multiple researchers using different analytical techniques across several separate studies conducted since the 1970s. But it's worth being equally clear about a separate, more recent line of research that complicates a simple, this confirms a real crucifixion reading of that blood evidence. A 2018 study led by forensic researchers Matteo Barini and Luigi Garlascelli, using modern bloodstain pattern analysis techniques of the kind used in criminal forensics, examined the actual flow patterns and directions of the bloodstains on the Shroud and found them inconsistent with a single static body position, of the kind a real body wrapped in a burial cloth would have produced. The stains, under this specific forensic analysis, appeared more consistent with an image deliberately or artificially created in multiple separate applications, rather than with blood flow from a genuine single wounded body lying in one consistent position. The specific methodology behind this finding is worth understanding briefly, because it's a genuinely clever application of an established forensic technique to a historical question. Bloodstain pattern analysis, routinely used in modern criminal investigations to reconstruct how a violent incident actually occurred based on the direction, shape, and flow pattern of blood found at a scene, works from a well-established principle. Blood flowing from a wound under the influence of gravity follows a predictable, physically consistent pattern, depending on the exact position and orientation of the body at the time. When Barini and Garlascelli applied this same analytical framework to the Shroud's bloodstains, they found the flow directions and patterns didn't consistently match what a real body, in any single realistic burial or crucifixion position, would actually have produced. Some stains appeared to flow in directions inconsistent with the specific body position the rest of the image otherwise depicts. This finding doesn't definitively settle the broader authenticity question on its own either, and it has itself drawn some methodological pushback from other researchers who have questioned certain aspects of the study's assumptions about exact body positioning during image formation. But it's a genuinely important, directly relevant piece of the scientific record that a fair, balanced treatment of the blood evidence needs to include alongside the earlier findings, confirming the stains contain real blood components, rather than presenting the "it's real blood" finding in isolation as though it settles the broader question of how that blood came to be on the cloth in the first place. Put all of this together, and here's an honest, balanced summary of where the actual body of scientific evidence currently stands. The image formation mechanism on the shroud remains a genuinely interesting, not fully and conclusively resolved technical question, though multiple plausible mechanisms, including various proposed contact transfer and chemical degradation processes achievable with medieval-era materials and techniques, have been proposed in the peer-reviewed literature, even though no single explanation has achieved complete universal consensus among researchers who study the object. Researchers have specifically explored techniques involving heated bas-relief templates combined with natural pigments that could degrade over centuries in ways that mimic some of the image's unusual properties, along with other proposed mechanisms involving chemical reactions between organic substances and the linen fibers themselves. Genuine, testable hypotheses that haven't been definitively proven, but that at minimum demonstrate plausible medieval-era pathways exist, even if none has yet achieved the kind of decisive, universally accepted experimental replication that would fully close the question. The 2015 DNA study found genuinely diverse genetic material on the cloth's surface, but the research team's own stated conclusion favored contamination from centuries of extensive handling as the most scientifically responsible explanation, not evidence of ancient travel along historical trade routes. The pollen evidence remains real, but scientifically contested, complicated by legitimate concerns about the original researchers' broader methodological credibility. And the 1988 radiocarbon dating, still the single most direct and rigorously controlled age determination test ever performed on the cloth, places its origin in the medieval period, a conclusion that a minority reweaving hypothesis has challenged but not displaced as the prevailing scientific position. None of this proves the shroud is definitively a medieval creation with total certainty, and responsible scientists studying it have generally been careful not to claim that level of absolute certainty either, particularly given the genuinely unresolved question of exactly how the image itself was produced. But it's equally important, and arguably more important, to be clear that the actual body of evidence doesn't support the more dramatic, popularly circulated claims either, that the DNA evidence proves an ancient Silk Road journey, or that mainstream radiocarbon dating has been scientifically debunked and discredited. The real, complete picture is more measured and, in its own way, more scientifically honest than either the most enthusiastic authenticity claims or a simple dismissal would suggest: a genuinely old and extensively studied object, carrying a real, still partially unexplained image formation mystery, tested by rigorous, mainstream, peer-reviewed radiocarbon dating that points toward a medieval origin, with several additional secondary lines of evidence that remain genuinely contested, complicated, or, in the specific case of the widely cited DNA study, actually pointing toward a considerably more mundane explanation than the more sensational popular retellings tend to present. There's a broader lesson in how this particular story has been told and retold over the years, worth naming directly, because it applies well beyond just the Shroud of Turin specifically. A real peer-reviewed scientific study, conducted by real researchers at a real university and published in a real, reputable journal, can still end up being seriously misrepresented in popular retelling, sometimes by quietly leaving out the researcher's own stated conclusions and replacing them with a more dramatic, more narratively satisfying alternative interpretation the original authors never actually proposed. The 2015 DNA study is a genuinely clean example of exactly this pattern. Real data, real peer review, real scientific rigor in the collection and sequencing process, paired with a study conclusion that gets quietly dropped or reversed in less careful retellings because the cloth was probably contaminated by centuries of handling, makes for a considerably less dramatic story than the DNA proves an ancient journey along the Silk Road, even though only one of those two framings is what the actual scientists who did the actual work actually concluded. There's something worth sitting with in that, beyond even the specific facts of this one particular object. The most honest version of any scientific story is rarely the most dramatic one, and learning to notice when a popular retelling has quietly swapped a researcher's own careful, hedged conclusion for a more exciting alternative is a genuinely useful habit, whether the subject is a 2,000-year-old cloth or any other scientific finding making the rounds online. The real Shroud of Turin story doesn't need embellishment to remain genuinely fascinating. An object this extensively tested, this carefully studied, and still carrying at least one genuinely unresolved technical mystery at its center, earns its place in the conversation on the strength of what's actually known, without needing anyone to oversell what the evidence doesn't actually say. If this gave you a more accurate, more balanced picture of what the Shroud's DNA testing and the broader body of scientific research surrounding it actually shows, tell me in the comments which part surprised you most. The DNA researchers' own contamination conclusion, the credibility concerns around the pollen research, or the 2018 blood stain analysis complicating the earlier blood findings. I read every comment, and it's usually where the next deep dive comes from. And if you want more of the real, evidence-based story behind the world's most enduring mysteries, subscribe now. We're just getting started.